Today is March 19th, 2021. It's the first day of our new unit, Unit 7. Unit 7 is all about genetics. So we're going to provide a brief introduction to genetics today, and then we're going to start talking about and defining some really important terms that are going to set the tone for this unit. This is one of the most important units in the class. In fact, the next 3 units, Unit 7, Unit 8, and Unit 9, are going to be the majority of your EOC exam in May. So if you have not been in the habit of taking notes at home, now is the time to do it. If you have not been in the habit of participating, now is the time to do it. If you have not been in the habit of turning in assignments on time and doing the warm-ups and exit tickets every day, I promise you, now is the time to do it. The EOC exam is going to touch on these concepts quite heavily. This is where we put in our best work. This is where we get the biggest bang for our buck. So I really want to emphasize the importance of note-taking, the importance of asking questions if and when you are confused. There's absolutely nothing wrong with being confused. We are going to talk about some terms and concepts that will be new to you all. So I expect that there will be some confusion, and I'm happy to clarify anything and anywhere I can. That being said, today's focus will be on an introduction, a brief overview of genetics. We're going to review meiosis a bit. So the objective is to explain the connection between meiosis, protein synthesis, and genetics. The essential question, how does genotype affect phenotype? So we see a couple terms there that we should be familiar with: meiosis, protein synthesis. We might be kind of familiar with genetics. I think we've heard that term tossed around in this class before as well. But genotype and phenotype are probably new to us. We will define those terms and several others today. All right, so for my folks at home, I'm hoping and trusting that you have notebooks out, pen and or pencil in hand, highlighter sitting nearby, whatever you do to take some nice, copious, organized notes. I hope you're doing it. And to my 2 people here, I've got Lisa here with me today, as well as Sayana. Looks like they're ready to go. So we're going to go ahead and move forward. You all are my indicators in terms of how fast I should be going, okay? So we're following your pace today. Okay, so before we really talk about genetics, we need to be clear about the definition of inheritance. Socially, inheritance means what kind of assets are you getting from your older generations, your elders, your parents? What are they leaving you when their time to move on has come? In biology, inheritance refers to similar things. You're being passed on something from your parents. In this case, we're not talking about a house or a car or money or stocks or equities. We're talking about genetic material, probably more important than all the other things that I just mentioned. So inheritance is the process by which genetic material is passed on from parent to child. The genes you receive from your parents determine most of your traits. Most of. The environment is also going to play a role, and we will talk more about that throughout this unit, and we've talked about it in the past as well. But many of your traits are heavily influenced by your genes. All right, before I move on, what is a gene? Does anybody remember what a gene is? Hopefully you all do. What is a gene? Not the denim. Not an example, but just the definition. Okay, so a gene can determine a trait. So, for example, a gene can determine your eye color. Good. So you could have blue eyes. There's a gene that could give you blue eyes. Abigail, I always want to call you Michelle because I just read the name. Abigail gives us basically the best definition in the chat. A gene is just a piece of DNA. It's a short segment of DNA. It's like a chapter in a book or a word in a sentence or a letter in a word. We've got 3 billion A's and T's and C's and G's that make up our human DNA. Within those 3 billion letters, we've only got about 30,000 genes, sections of DNA that actually code for a protein. That's really the most important connection we've got to make. Genes are pieces of DNA, as Abigail puts it, but they code for proteins. They allow us to make something. All right, just want to be clear about that. So let's throw it back to meiosis. We're going to have, I think, over the course of the next 3 slides, 5 sentences that will be missing some words. Let's see if we can fill in these words. The first one, meiosis is the process by which blank are created to be used for blank reproduction. What should be number 1 there? What's created by meiosis? Excellent. Thank you, Janelle. Meiosis is the process by which gametes are created, and those gametes then get used for what? What kind of reproduction? Thank you, Lisa. Sexual reproduction. If you are unclear about this, or if you have not written this down in the past, I would strongly encourage you to write these sentences down, or at least to capture the gist of them somewhere in your notes, summarize them, paraphrase them. But we've got some important information about meiosis here. Meiosis continues to be one of those standards that we struggle with. For those of you who were successful in uploading your student data trackers yesterday, I appreciate that. And what I'm seeing, of course, I've got all of the aggregate data from both of my biology classes. We are struggling with meiosis. So we wanted to make sure that we put these slides in to go over it a little bit. All right, the next sentence. A primary purpose of meiosis is to create blank cells, which have half the number of blank of the body cells. So what are we trying to create? What kind of cells have half the number of blank? What's that? That is going to come up later. JT, your word, unique, is going to come up later. But again, Janelle gives us the correct answer here. Haploid. That's why we say they have half the number of blank. What do they have half the number of? Okay, you're thinking about the actual number. But what is with 46 what? Humans have 46 chromosomes. Good. That's what we're looking for. And Abigail gives it to us in the chat as well. So I'm going to just read both of these sentences now that all of the words have been revealed. Meiosis is the process by which gametes are created to be used for sexual reproduction. A primary purpose of meiosis is to create haploid cells, which have half the number of chromosomes of the body cells. Okay, there's a lot of information packed in those 2 sentences. Any questions about that, though? Sion, you feel good about this? Okay. This is a throwback to Unit 3 when we talked about the cell cycle. I'm going to leave this up just a little bit longer so anybody who needs to can capture this information. Meiosis is the process by which gametes are created to be used for sexual reproduction. A primary purpose of meiosis is to create haploid cells, which have half the number of chromosomes of the body cells. Before the chromosomes split during the cell cycle, homologous chromosomes exchange part of their DNA. What's that process called? And we can see it in both of the images on the screen. What's that process called when they exchange their DNA? Or just a small piece of it? In fact, you can kind of see it in the little GIF on the left side. Good, thanks, JT. Oh, sorry, everybody came in. Thanks, Abigail and Melissa as well. So this process is called crossing over. They're just going to exchange just a little piece of a small piece of their DNA. Does anybody remember why they do that? What's the benefit of doing that? That doesn't come up on the screen, but I just wanted to ask another question. Why do cells do crossing over? Okay, this is something we might need to continue thinking about. Crossing over provides the benefit of genetic diversity. So we're just kind of scrambling the DNA even more than it already is. And this makes it more likely that we create more genetic diversity. Okay, that's the whole point of crossing over. The second sentence says, as the chromosomes separate during anaphase 2, genes are randomly separated into how many daughter cells? 4, excellent. And are they identical or are they unique? They're unique, good. So genes are randomly separated into 4 unique daughter cells. Oh, I thought it was going to say unique. It says haploid. Forgive me. Okay, I'm leaving this up for a little bit just in case anybody wants to capture some of this information. Okay, last sentence. The gametes blank contain only one set of chromosomes, which is half of the parent's DNA. I'm just going to show that one because it's kind of not obvious. But of course, gametes are egg and sperm cells. They are the sex cells. Remember, sexual reproduction requires blank to make offspring. So what's happening on the screen in that animation? What's that process called? Good, thank you, Lisa. This is fertilization. So it's an important distinction to make. Meiosis is not fertilization. They're not the same thing. Meiosis makes fertilization possible because meiosis produces sex cells. But fertilization doesn't happen until those 2 different sex cells are actually joined. We can see that happening on the screen. The sperm cells are colliding with the egg cell and trying to penetrate its protein covering. Once it does, that's when fertilization takes place. All right, good job. So nothing to write here. I just want to kind of depict some of the genetic diversity that comes out of sexual reproduction. Obviously, we all have 2 biological parents. And obviously, those 2 people also have 2 biological parents. So your grandparents gave half of their chromosomes to your parents. And we can see it really represented here with colors. Mom gets a little bit of her mother's DNA and a little bit of her father's DNA. Dad gets a little bit of his mother's DNA and a little bit of his father's DNA. Then, of course, when they combine and fertilize and eventually they have an offspring that becomes you get a little bit of their DNA. But if we look at the colors, it's pretty conveniently shown here. It depicts just how much you're getting from each of your 4 grandparents. And so this process of fertilization It's showing you how genes are being passed on. DNA is being passed on from one generation to the next. And this is what genetics studies. It's the study of heredity. Okay. Boom. One more or a couple more definitions here. Genetics is the study of heredity, aka passing on genes/traits from parents to offspring. Every individual inherits 2 copies of each gene because we each have 2 parents. You get 1 copy from one parent and you get another copy from another parent. The fancy scientific word for these copies is an allele. It's a weird word to say. A-L-L-E-L-E. Allele. So of course we can look on the right side of the screen here and we see different traits. There's a trait for hair color. There's a trait for blood type. There's a trait for eye color. There's a trait for growth. Many of the traits that make us who we are actually determined by a singular gene. And that gene makes a singular protein that contributes to whatever the trait is. But we're getting copies of that gene from both of our parents. And those copies will combine in unique ways, which we will see soon. We represent alleles with letters. The letters themselves are arbitrary. They actually don't really have anything to do with the gene. So we can choose whatever letter works for us. If we're talking about hair color, we might choose the letter H. If we're talking about blood type, there are actually specific letters for that, which we'll talk about later in the unit. If we're talking about eye color, we might choose the letter E. Okay, so whatever we feel comfortable using, unless it is something like blood type, or unless we're talking about a sex-linked trait, we'll talk about those 2 things next week, then we can choose whichever letter we want to use. All right, we'll get some practice with this today and for the rest of the unit. This unit is going to take us all the way through spring break, so we've got some time. Okay, so there are 2 types of alleles. There is a dominant allele, which we represent with a capital letter. And there are also recessive alleles, which we represent with lowercase letters. I'm going to draw on the board a chart, or a table rather, that I would definitely encourage you all to copy down in your notes. Okay, so one of my 2 people who are actually here in person, How do I represent a dominant allele? It's on the screen. A capital letter, yeah. In this case, I'm going to use the letter E. Is that okay with you all? Okay, I'm just going to use the letter E. And then how do I represent a recessive allele? The lowercase, yeah. In fact, just to make it a little bit more obvious, does this green work? I'm going to use different colors as well. Okay, so the most important thing on this slide to remember is that the dominant allele is stronger than the recessive allele. That's why we use the word dominant. What this means is that if they both appear, remember, we're getting one allele from each parent. So if they both appear in a combination, the dominant allele takes precedence. The dominant allele is what is actually going to be expressed. Okay, so remember, each of these alleles is a copy of a gene. And what that means is that they each code for a specific type of trait. You might have tall versus short. You might have round versus square. You might have black versus white. So each copy of the gene is coding for a specific trait. The dominant allele codes for the dominant trait. The recessive allele codes for the recessive trait. Dominant alleles hide recessive alleles. That just means that if they're both present, if I've got one allele that is dominant and another that is recessive, the dominant allele is going to determine the trait. We'll talk more about that, all right? These terms are going to be a little bit confusing as we first get introduced to them, but I promise they'll make sense pretty quickly, probably by the end of today. Okay, so again, we're getting one allele from each parent. Your father gives you one allele, your mother gives you one allele. That means that you've got a combination of two alleles, and there are three possible outcomes from that. We could be what is called homozygous dominant. Homozygous dominant shows us that we've got two capital letters. Two capital letters. Okay, I'm going to write that in my table now. Now, homo is a prefix that means same. So homozygous means that you've got two zygotes that are the same. Either they're both dominant, you've got two dominant alleles, or you've got two recessive alleles, but they're both the same. That's why we use the prefix homo. Another way of saying homozygous is true breeding. Keep that in mind as well. True breeding is another way of saying homozygous. We also see, therefore, I'm going to jump to number 3, homozygous recessive is two lowercase alleles. They are the same and they're both lowercase. Again, I would really recommend copying down this table. It's going to become quite helpful to you for what we do on Monday. So I would definitely recommend it. All right. And then, of course, if we have one dominant allele and one recessive allele, we've got one of each, that means that they are different. They look different. One is capital, one is lowercase. They are different. One gives you, or one codes for the dominant trait, one codes for the recessive trait. They're different. That's why we call them heterozygous. Hetero is a prefix meaning different. Keep in mind, though, that the dominant allele masks the recessive allele. And that just means that the dominant allele takes precedent over the recessive allele. If they are both present, as is the case in a heterozygous genotype, or in a heterozygous combination, I should say, that dominant allele is going to determine the trait. We'll talk more about that. My favorite thing to say. Oh, that's not what I wanted to do. One thing I want to mention to you all here. Probably can't hear me. Typically, we do like to write the capital E before the lowercase e. That's just kind of like the correct naming convention. But nothing is incorrect about writing the lowercase e first. That's not wrong. We're focused on the combination of the two. It doesn't necessarily mean anything that you write the capital letter first. Okay, so we could also have so there's nothing wrong with writing that one right there, lowercase e, capital E. They're the same thing. We're looking at the comment. It's like saying 3 plus 2 is 5, but 2 plus 3 is also 5. It doesn't matter which order you add them in. Does that make sense? All right. So we can see now our 3 possible combinations. I promise you, there are no others. We will not be talking about others in this class. These are the 3 combinations we need to know. Homozygous dominant, heterozygous, and homozygous recessive. Any questions about anything so far? And I just want to remind you that you might see this phrase true breeding coming up. It's literally a synonym for homozygous. All right, true breeding is just telling you this is a purebred. This person only has the same allele. It doesn't mean dominant, though. It just means homozygous. It could be homozygous recessive. Keep that in mind. All right, so let's practice. We've got the key that I wrote on the board. Hopefully you all have copied that into your notes. We can see these different combinations of alleles on the screen now. I want you all to tell me if they are HD, HR, or HE. HD, of course, being homozygous dominant, HR being homozygous recessive, and HE being heterozygous. I'm going to start with my 2 people who are here with me in person as they glare at me angrily. Sayana, with this first combination, I'm looking way on the far left, AA. You said HD? Excellent. How did you know that? Yes, the chart. Good. Okay, you guys, you're making my standards class not look very good. Of course, this is HD. Why? The first thing we need to look at, are the letters the same? Yes, they're both capital here. They look the same. They are the same. They code for the same trait. They're both dominant alleles. They're both capital letters. And then the second thing we really needed to determine, but I kind of just gave it away, are they capital or are they lowercase? They're both capital letters. That means that they are representing the dominant allele. So this is a homozygous dominant combination. Okay, coming to you now, Lisa. The second one, CC, what do we think? HE, wonderful. And how did you know that? Excellent. Yeah, we've got one of each here. We've got one capital letter, aka one dominant allele, and then we also have one lowercase letter, aka one recessive allele. So they're different in that case. The prefix that means different is hetero. All right, my two people who are here in person have proven themselves. All right, they're stamped. They're solid. So I'm looking for you all who are at home right now to get me the rest of the way. FF, what would that be? Send it to me in the chat.
HR.
Good. Wonderful. Thank you, JT, and everybody. Josue, Melissa, Dejan. Good job. It's HR. Next.
HE.
Good. We're seeing HE here. This is heterozygous because we've got one of each. One dominant, one recessive. What's next? And then Dejan, yep, thank you, HE, for the BB. It's a lot of letters being thrown around. Okay, DD.
HD.
That's HD, homozygous dominant. They are the same, and they're also both capitals, so they are homozygous dominant. What's next? HE, good. And then the last one, HH. What do you got for me, Lisa, for this last one? Excellent. And thanks to the folks at home for jumping on that one as well. HR. Good. So we're thinking this must not be all that hard, right? Okay, now we're going to jump in with some new stuff. I promise if you can just remember that basic stuff, though, we're going to be in a good place going forward. Two more definitions that I do want you to write down. One of my students in my last class, my standards class, asked me a great question, which I won't tell you exactly what the question was, but I'm going to allow that question to kind of dictate how I'm going to explain it this time so that the question doesn't come up again. As it says at the top of the screen, there are two ways to describe traits. We're describing it in the same way, just like we have different names. I refer to, or you all could refer to me. Well, no, there's only one way you guys can refer to me, Mr. Rudd. But I would answer to both Mr. Rudd and to my first name. The reason for this is because there are two ways of describing me. They're both the same way of describing me. We could think about this in terms of different languages. We could say bathroom in English or baño in Spanish. We're describing the same thing, but in two different ways. In either language, you know exactly what I'm talking about. And actually, it's baño in Turkish too, so there's a third way. Okay, anyway, genotype and phenotype are two different ways to describe the same thing. I just want to be clear about that. The genotype is the genetic description. It's the actual letters that we use to represent alleles. But it could also be words like homozygous dominant or heterozygous or homozygous recessive. Those are also genotypes or genotypic descriptions. The phenotype, on the other hand, is the physical description of the trait. This is what you can actually make out and see with your eyes. This could be something that's tall or blue or brown or heavy. We're using these vibrant, vivacious, descriptive words to describe what we're seeing. That's a phenotype. The genotype is literally just capital A, or heterozygous, or homozygous recessive. That's the genotype. But there are two ways of describing the same thing. So the reason, are we done? The reason I chose to use the letter E here, I want to talk about this trait, the trait of attached or detached earlobes. If you've got an earlobe that dangles a little bit and there's a little bit of separation between your earlobe and the side of your face, you've got a detached earlobe. Some people have attached earlobes. Believe it or not, Detached earlobes are dominant traits. So most people in the population will have detached earlobes, but I'm sure at least one person in the class has has attached earlobes. It's not a bad thing. It doesn't mean that you're sick or anything like that or weird. It just means you have the recessive trait. So here's how we would kind of write this. 1 second. Tilt my screen up a little bit. Detached earlobes are dominant to attached earlobes. That's how we would describe this trait. We could also go the other way. We could also say attached earlobes are recessive to detached earlobes. It's the same thing. That's like saying Shaquille O'Neal is taller than Mr. Rudd versus Mr. Rudd is shorter than Shaquille O'Neal. The same thing. We're describing the relative heights. In this case, we're describing the relative outcomes of this trait. Detached earlobes are dominant to attached earlobes. Okay. That's why I use the letter E, right? Because we're talking about ears now. So which one is the dominant trait? Detached or attached? Which one is the dominant trait? Based on that sentence. Detached, yeah. So that means that my dominant allele, the capital E, is coding for detached earlobes. What is the recessive trait then? Obviously. Attached earlobes. And I'm pronouncing it weird just so that there's clear distinction between the two. All right, so my lowercase e, my recessive allele, is coding for attached earlobes. My dominant capital E is coding for detached earlobes. All right, now we don't just end up with one allele though. That's kind of the problem here. We get two alleles, one from dad, one from mom. So we need to think about what are the outcomes of the combinations that we already talked about. Homozygous dominant, heterozygous, homozygous recessive. What trait are we actually going to get from those combinations? So let's start with one that should be pretty easy. If I'm homozygous dominant, then which trait am I going to end up with? Detached or attached? If I'm HD, homozygous dominant. Yes. If I'm homozygous dominant, HD, then which of these actual phenotypes am I going to end up? Detached. Homozygous dominant. Of course, homozygous dominant has two dominant alleles. So the only allele that I'm getting is for the dominant trait. I don't even have an allele for the recessive trait. There's nothing in my DNA that would ever make the recessive trait. I can only get detached ears. All right, so homozygous dominant, of course, that genotype gives me the phenotype of detached earlobes. Describing the same thing, though. They're describing the same thing. All right. Now let's think about this guy or gal or person down here. What does that say? Homo recessive, homozygous recessive. What does that genotype get me? Which phenotype does that genotype get me? Detached or attached earlobes? If I'm two lowercase e's, which phenotype am I going to get? Yeah, I'm going to get attached earlobes. I'm trying to make it easier for you all with the colors here. How do we feel about this, Sana? Good? Good? Okay, excellent. And last one, this one can be a little bit tricky. It's a little bit confusing, but now we want to think about the heterozygous genotype. I've got one of each with the heterozygous genotype. What phenotype do I get from that? Detached or attached? And no, I don't have my left ear detached and my right ear attached. That's not how it works. Okay, Abigail, would you mind explaining how you, or like, why do we know that it's detached? You are right, but can you explain that?
Because you said that the dominant was gonna dominate.
Right, thank you. It's pretty simple. The dominant allele is going to dominate. So whatever trait that the dominant allele is coding for, in this case, it's coding for detached earlobes. If the dominant allele is present, it's going to take over. It will determine the trait. Anytime I see the dominant allele, I'm going to have the dominant trait. Okay, so let me ask some questions then. How many genotypes will give me the dominant allele? I'm sorry, how many genotypes will give me the dominant trait? Good, excellent, Dijon. 2 genotypes will give me the dominant trait. Homozygous dominant gives me the dominant trait, and heterozygous also gives me the dominant trait. But how many genotypes will give me the recessive trait? Good. There's only one genotype that can give me the recessive trait. That's homozygous recessive. Okay. Might be hard to see. Only one genotype can possibly give me the recessive trait. In order to have, this is going to be important as we move through this unit. In order to have the recessive trait, I have to be homozygous recessive. That's the only way I can get the recessive trait. Because if I'm heterozygous, I've got a dominant allele and it's going to give me the dominant trait. All right, so if you can remember that, you are in a great place as we move forward in this unit. The only way to get the recessive trait is if I'm homozygous recessive. If I've got 2 recessive alleles, 2 lowercase letters. All right, we're going to practice with this so much, I promise you, it's going to be sickening. But you guys are going to get good at it. All right, genotype versus phenotype. Here are some pictorial representations of the differences between the two. Genotype is referring to the actual genes, the alleles that are present. You get 1 copy from each parent. Phenotype is what can I see? What are the traits that I can actually visualize? They're describing the same thing, though. They're just 2 different ways of doing it. We will talk more about this, but of course, not everything is as simple as, oh, you get 1 dominant or you get one recessive. You either have the dominant trait or you have the recessive trait. There's no in between. That's obviously not the real world. We have huge variations that are present throughout the 8 billion people who live on this planet. Those variations come because there are multiple genes that determine traits. We call these polygenic traits. Yes, there are some traits, like I just talked about, detached or attached earlobes, that are determined by a single gene. Those traits do exist. But many of our traits are determined by multiple genes. This is why we have a lot of variation. Imagine if we lived in a world where there was a tall gene, there was a tall trait and there was a short trait. If you got the tall trait, you were 7 feet tall. If you got the short trait, you were 4 feet tall. And everybody was the exact same height. All the tall people were 7 feet. All the short people were 4 feet. Or hair texture. Either you had perfectly straight hair or your hair was extremely coiled. We know that there are 4B and 3B and 2B and 2A and 3C. At least that's how I understand it. I don't know that much about hair, but I'm sure somebody out there does. So I know there are different hair textures, though. So it's not just perfectly straight or perfectly coiled. There's a mixture. Okay, and these are because many of our traits, especially the ones that we can easily see, are determined by multiple genes. All right, now I want you guys to tell me, is the description a genotype or a phenotype? So number one, curly hair, would that be a genotype or a phenotype? Good, that's a phenotype. The way I like to think about it too, pheno refers to physical characteristics, pH and phenotype, pH and physical characteristics. All right, what about AA? What is that, genotype or phenotype? Yeah, that's geno. What about greenish brown? That's a phenotype as well. It's describing something we can actually see with our eyes. What about XY? This would be a geno. Now, this one you're looking like, wait a minute. This whole time we've only been talking about combinations of the same letter. Either it's just A, or it's just X, or it's just Y, but not X and Y. We'll talk more about that next week. X and Y refer to our sex chromosomes. You guys have probably heard me say that before. But there are some examples and some types of inheritance that we'll talk about in which we'll be using different letters. So it might feel easy now. If you haven't written this chart down, I definitely encourage you to do so, because we are going to layer on, okay? We're providing the foundational level here. And then last one, spotted. Is that a genotype or a phenotype? That's pheno. Good. I feel like I'm saying an Italian word, pheno. Okay. So my top example says that purple is dominant to white. Purple is dominant to white. I'm given on the left side 3 genotypes. And my job now is to figure out the phenotypes that correspond to those genotypes. All right. Purple is dominant to white. That's the same thing as saying white is recessive to purple. Same thing. Okay. So my first genotype, what is that genotype called? Number 1. What's that? What kind of dominant? Yes, it does give it. You're right. It does give us that. I don't want to say it out loud yet. You're right, Lisa. But what is the actual name of that genotype? Good. That is homodominant. It's HD. So number 1 is the homozygous dominant genotype. Good job. And Lisa already gave me the answer, but I want to hear it from somebody else. What phenotype does homozygous dominant give us in this case? Purple. Yeah, because purple is the dominant trait. Good job. All right, let's jump down to number 3. I'm skipping number 2. What's the name of the genotype in number 3? Lowercase p, lowercase p. What's that genotype called? Good. Thank you, Melissa. Thank you. Ooh, wait a minute. I'm skipping to number 3. Lowercase, lowercase. Lowercase, lowercase. Yes, HR, HR. Good. Have you ever seen that? Never mind. Never mind. Lowercase p, lowercase p. That's homozygous recessive. Good. I just gave it away. But of course, it gives us the phenotype white. I distracted myself. You guys made me think of this comedian joke, but I don't think it's appropriate. So the phenotype is indeed white here. And then number 2, what's the name of that genotype? Capital P, lowercase p. What's that called? Good. This is the HE. I'm never going to be able to say HE again without thinking about this comedian. So you guys have ruined it for me. What's the phenotype that I get from heterozygous? Purple or white? Good. We get the dominant phenotype when I have the heterozygous genotype. I still get the dominant phenotype, so it's purple. Are we good on this? How do we feel? Good. All right. Okay, so Sayana, number 1. We're given the phenotype on the right side. We're told that light hair is recessive to dark hair. Another way to put that is dark hair is dominant to light hair. We're given 3 phenotypes on the right side. I want you to tell me the genotype for number 1. There are 2 possible answers, technically, but which one do you want to go with? Oh, sorry. The letter we're using is H. Good question. Oh, you tell me, is it capital or lowercase?
Capital.
Good. What's next? Because it's also dark. Wonderful. Excellent. Good job. Thank you. So I think we're catching on here pretty quickly. There are 2 ways to get the dominant trait. You could be homozygous dominant or you could be heterozygous. This is why more people have the dominant trait, because it's easier to get it. There are 2 different ways to get it. All right. So you all have 20 minutes, 22 minutes. That's a lot of time to knock out the exit ticket as well as the asynchronous assignment. I would definitely encourage you to do them both. That way you don't fall behind. Nobody likes being behind because then you got to catch up and you're stressed out. At least that's how I feel. So just use the time that you're given to knock out both of these things. And there were a few of you who showed up a little bit late. Please make sure that you do the warm up as well. It's Unit 7, Day 1. So the assignment is called U7D1, Basic Genetics Practice. Go ahead and knock that out for me, folks. I'll be here on mute if there are any questions. All right, folks, everybody have a great weekend. Let me know if you need anything between now and Monday. I hope you get some time to relax, and I hope you were productive today. Good job, good job, good job. See you, JT. Thank you all.